Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Macromolecule. 73 languages. Chemical structure of a polypeptide. Macromolecule. EU-Park definition. Macromolecule. Large molecule. A molecule of high relative molecular mass, the structure of which essentially comprises the multiple repetition of units derived, actually or conceptually, from macromolecules. Redirects here. For the journal, see Macromolecules, Journal. Macromolecular chemistry. Redirects here. For the journal formerly known as Macromolecular Chemistry, see Macromolecular Chemistry and Physics. This article needs additional citations for verification. Please help improve this article by adding citations to reliable sources. Unsourced material may be challenged and removed. Find sources: Macromolecule News Times, Newspapers Times, Books Times, Scholar Times, Chester, September 2024. Learn how and when to remove this message. A macromolecule is a very large molecule, important to biological processes, such as a protein or nucleic acid. It is composed of thousands of covalently bonded atoms. Many macromolecules are polymers of smaller molecules called monomers. The most common macromolecules in biochemistry are biopolymers, nucleic acids, proteins, and carbohydrates, and large non-polymeric molecules, such as lipids, nanogels and macrocycles. 1. Synthetic fibers and experimental materials such as carbon nanotubes. 2, 3, are also examples of macromolecules. Definition, edit. The term macromolecule, macro plus molecule, was coined by Nobel laureate Hermann Staudinger in the Explore Our Developer-Friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Molecules of low relative molecular mass. Notes. 1. In many cases, especially for synthetic polymers, a molecule can be regarded as having a high relative molecular mass if the addition or removal of one or a few of the units has a negligible effect on the molecular properties. This statement fails in the case of certain macromolecules for which the properties may be critically dependent on fine details of the molecular structure. 2. If a part or the whole of the molecule fits into this definition, it may be described as either macromolecular or polymeric, or bipolymer used adjectivally. 4. 1920s, although his first relevant publication on this field only mentions high molecular compounds in excess of 1,000 atoms. 5. At that time, the term polymer, as introduced by Bertillus in 1832, had a different meaning from that of today. It simply was another form of isomerism, for example with benzene and acetylene, and had little to do with size. 6. Usage of the term to describe large molecules varies among the disciplines. For example, while biology refers to macromolecules as the 4 large molecules comprising living things, in chemistry the term may refer to aggregates of 2 or more molecules held together by intermolecular forces, rather than covalent bonds but which do not readily dissociate. 7. According to the standard EU-Park definition, the term macromolecule as used in polymer science refers only to a single molecule. For example, a single polymeric molecule is appropriately described as a macromolecule or polymer molecule rather than a polymer, which suggests a substance composed of macromolecules. 8. Because of their size, macromolecules are not conveniently described in terms of stoichiometry alone. The structure of simple macromolecules, such as homopolymers, may be described in terms of the individual monomer subunit and total molecular mass. Complicated biomacromolecules, on the other hand, require multifaceted structural description, such as the hierarchy of structures used to describe proteins. In British English, the word macromolecule tends to be called high polymer properties, edit. This section needs additional citations for verification. Please help improve this article by adding citations to reliable sources in this section. Unsourced material may be challenged and removed. May 2013. Learn how and when to remove this message. Macromolecules often have unusual physical properties that do not occur for smaller molecules. How? Another common macromolecular property that does not characterize smaller molecules is their relative insolubility in water and similar solvents, instead forming colloids. Many require salts or explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. DNA RNA proteins. Encodes genetic information, yes yes no. Catalyzes biological reactions, no yes yes. Building blocks, type, nucleotides amino acids. Building blocks, number, 4420. Strandedness double single. Structure double helix complex. Stability to degradation high variable. Repair systems yes no. Particular ions to dissolve in water. Similarly, many proteins will denature if the solute concentration of their solution is too high or too low. High concentrations of macromolecules in a solution can alter the rates and equilibrium constants of the reactions of other macromolecules, through an effect known as macromolecular crowding. 9. This comes from macromolecules excluding other molecules from a large part of the volume of the solution, thereby increasing the effective concentrations of these molecules. Linear biopolymers, edit. All living organisms are dependent on 3 essential biopolymers for their biological functions: DNA, RNA and proteins. 10. Each of these molecules is required for life, since each plays a distinct, indispensable role in the cell. 11. The simple summary is that DNA makes RNA, and then RNA makes proteins. DNA, RNA, and proteins all consist of a repeating structure of related building blocks, nucleotides in the case of DNA and RNA, amino acids in the case of proteins. In general, they are all unbranched polymers, and so can be represented in the form of a string. Indeed, they can be viewed as a string of beads, with each bead representing a single nucleotide or amino acid monomer linked together through covalent chemical bonds into a very long chain. In most cases, the monomers within the chain have a strong propensity to interact with other amino acids or nucleotides. In DNA and RNA, this can take the form of Watson-Crick base pairs, GC and a T or a U, although many more complicated interactions can and do occur. Structural features, edit. Because of the double-stranded nature of DNA, essentially all of the nucleotides take the Form of Watson-Crick. Base pairs between nucleotides on the two complementary strands of the double helix. In contrast, both RNA and proteins are normally single-stranded. Therefore, they are not constrained by the regular geometry of the DNA double helix, and explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. So fold into complex three-dimensional shapes dependent on their sequence. These different shapes are responsible for many of the common properties of RNA and proteins, including the formation of specific binding pockets, and the ability to catalyze biochemical reactions. DNA is optimized for encoding information. Edit. DNA is an information storage macromolecule that encodes the complete set of instructions, the genome, that are required to assemble, maintain, and reproduce every living organism. 12. DNA and RNA are both capable of encoding genetic information, because there are biochemical mechanisms which read the information coded within a DNA or RNA sequence and use it to generate a specified protein. On the other hand, the sequence information of a protein molecule is not used by cells to functionally encode genetic information. 1.5. DNA has three primary attributes that allow it to be far better than RNA at encoding genetic information. First, it is normally double-stranded, so that there are a minimum of 2 copies of the information encoding each gene in every cell. Second, DNA has a much greater stability against breakdown than does RNA, an attribute primarily associated with the absence of the 2-hydroxyl group within every nucleotide of DNA. Third, highly sophisticated DNA surveillance and repair systems are present which monitor damage to the DNA and repair the sequence when necessary. Analogous systems have not evolved for repairing damaged RNA molecules. Consequently, chromosomes can contain many billions of atoms, arranged in a specific chemical structure. Proteins are optimized for catalysis. Edit. Proteins are functional macromolecules responsible for catalyzing the biochemical reactions that sustain life. 1. 3 proteins carry out all functions of an organism, for example photosynthesis, neural function, vision, and movement. 13. The single-stranded nature of protein molecules, together with their composition of 20 or more different amino acid building blocks, allows them to fold into a vast number of different 3-dimensional shapes, while providing binding pockets through which they can specifically interact with all manner of molecules. In addition, the chemical diversity of the different amino acids, together with different chemical environments afforded by local 3D structure, enables many proteins to act as enzymes, catalyzing a wide range of specific biochemical transformations within cells. In addition, proteins have evolved the ability to bind a wide range of cofactors and coenzymes, smaller molecules that can endow the protein with specific activities beyond those associated with the polypeptide chain alone. RNA is multifunctional. Edit. RNA is multifunctional. Its primary function is to encode proteins, according to the instructions within a cell's DNA. 1.5 They control and regulate many aspects of protein synthesis in eukaryotes. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. RNA encodes genetic information that can be translated into the amino acid sequence of proteins. as evidenced by the messenger RNA molecules present within every cell, and the RNA genomes of a large number of viruses. The single-stranded nature of RNA, together with tendency for rapid breakdown and a lack of repair systems, means that RNA is not so well suited for the long-term storage of genetic information as is DNA. In addition, RNA is a single-stranded polymer that can, like proteins, fold into a very large number of three-dimensional structures. Some of these structures provide binding sites for other molecules and chemically active centers that can catalyze specific chemical reactions on those bound molecules. The limited number of different building blocks of RNA, 4 nucleotides vs. 20 amino acids in proteins, together with their lack of chemical diversity, results in catalytic RNA, ribozymes being generally less effective catalysts than proteins for most biological reactions. The major macromolecules. Macromolecule. Polymer. Building block. Monomer. Bonds that join them. Proteins, amino acids, peptide. Nucleic acids, phosphodiester. DNA. Nucleotides, a phosphate, ribose, and a base adenine, guanine. Thymine, or cytosine. RNA. Nucleotides, a phosphate, ribose, and a base adenine, guanine. Uracil, or cytosine. Polysaccharides, monosaccharides, glycosidic. Lipids. Unlike the other macromolecules, lipids are not defined by chemical structure. Lipids are any organic, nonpolar molecule. Some lipids are held together by ester bonds, some are huge aggregates of small molecules held together by hydrophobic interactions. Carbohydrates, carbon, hydrogen, and oxygen. Major protein complexes. Branched biopolymers. Edit. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Raspberry L-agitanin, a tannin. Composed of core of glucose units. Surrounded by gallic acid esters and L-agic acid units. Structure of an example. Polyphenylene dendrimer. Macromolecule. 14. Carbohydrate macromolecules, polysaccharides, are formed from polymers of monosaccharides. 11 Because monosaccharides have multiple functional groups, polysaccharides can form linear polymers, for example cellulose, or complex branched structures, for example glycogen. Polysaccharides perform numerous roles in living organisms, acting as energy stores, for example starch, and as structural components, for example chitin in arthropods and fungi. Many carbohydrates contain modified monosaccharide units that have had functional groups replaced or removed. Polyphenols consist of a branched structure of multiple phenolic subunits. They can perform structural roles, for example lignin, as well as roles as secondary metabolites involved in signaling, pigmentation and defense. Synthetic macromolecules, edit. Some examples of macromolecules are synthetic polymers, plastics, synthetic fibers, and synthetic rubber, graphene, and carbon nanotubes. Polymers may be prepared from inorganic matter as well as, for instance, in inorganic polymers and geopolymers. The incorporation of inorganic elements enables the tunability of properties and forward/or responsive behavior, as for instance in smart inorganic polymers. See also, edit. List of biophysically important macromolecular crystal structures. Small molecule. Soft matter. References, edit. 1 ABCDE Stria L, Bird J.M., Timochko J.L., 2002. Biochemistry, 5th ed. San Francisco, W.H. Freeman. ISBN 978-0-7167-4955-4. 2 Life cycle of a plastic product archived 17 March 2010 at the Wayback Machine. AmericanChemistry.com. Retrieved on 1 July 2011. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 3 Gullaply, S. Wong, M.S. 2011. Nanotechnology, A Guide to Nano-Objects, PDF. Chemical Engineering Progress. 107, 528-32. Archived from the original, PDF, on 13 August 2012. Retrieved 28 June 2015. 4 Jenkins, A. D., Criticville, P. Steptoe, RFT, Suter, UW, 1996. Glossary of Basic Terms in Polymer Science, EU Park Recommendations 1996, PDF. Pure and Applied Chemistry. 68, 12 2287 2311. Do. 10.1351 /PAC199668122287. S2 SID 98774337. Archived from the original, PDF, on 2016-03. 04 retrieved 27 July 2013. 5 Staudinger, H. Fritschi, J. 1922. Über Isoprene und Kautschuk. 5. Mitteilung über die Hydrierung des Kautschuks und über seine Constitution. Helvetica Chimica Acta. 5, 5785. Do. 10.1004/aka.19220050050517. 6 Jensen, William B. 2008. The Origin of the Polymer Concept. Journal of Chemical Education. 85. 5624. Bibcode, 2008 JCHED 85624J. Do. 10.1021/ed085p624. 7 Van Hold, K.E. 1998, Principles of Physical Biochemistry Prentice Hall, New Jersey, ISBN 0-13. 720459-0. 8 Jenkins, A. D. Criticville, P. Steptoe, RFT. Suter, UW. 1996. Glossary of Basic Terms in Polymer Science, PDF. Pure and Applied Chemistry. 68, 12 2287. Do. 10.1351/PAC199668122287. S2 SID 98774337. Archived from the original, PDF, on 2007-02. 23. 9 Minton AP, 2006. How can biochemical reactions within cells differ from those in test tubes? J. Cell SCI 119, pt 14 2863 9 do 10.1242/JCS.03063 PMID 1682547 10 Berg, Jeremy Mark, Timochko, John L. Stryer, Lubert, 2010. Biochemistry, 7th ed. Biochemistry. Berg. W.H. Freeman and Company. ISBN 978-1-4292-2936-4. 5th edition available online through the NCBI bookshelf, link. 11 Walter, Peter, Alberts, Bruce, Johnson, Alexander S. Lewis, Julian, Raff, Martin C. Roberts, Keith. 2008. Molecular Biology of the Cell, 5th edition, extended version. New York, Garland Science. ISBN 978-0-8153-4111-6. 4th edition is available online through the NCBI bookshelf, link. 12 Golnick, Larry, Wheelies, Mark. 14 August 1991. The Cartoon Guide to Genetics. Collins reference. ISBN 978-0-06-273099-2. 13 Takes Miura, Masaharu, 2009. The Manga Guide to Molecular Biology. No Starch Press. ISBN 978-1. 59327-202-9. 14 Roland E. Bauer, Volker Enkelmann, Awel M. Wiesler, Alexander J. Bereshim, Klaus Mullen, 2002. Single Crystal Structures of Polyphenylene Dendrimers. Chemistry, a European Journal. 8, 17. 3858 3864. Do. 10.1001/1521-3765, 200209028 817.3.0.co, 2-5. PMID 122032880. External links, edit. Synopsis of Chapter 5, Campbell and Rees, 2002. Lecture notes on the structure and function of macromolecules. Several, free, introductory macromolecule-related internet-based courses archived 2011. 07-18 at the Wayback Machine. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Giant Molecules. By Ulysses McGee, ISSA Review Winter 2002-2003, ISSN 1542-9864. Cached HTML version of a missing PDF file. Retrieved 10 March 2010. The article is based on the book, Inventing Polymer Science, Staudinger, Carothers, and the Emergence of Macromolecular Chemistry by Yasu Fukawa. Categories: Macromolecules, Molecular Physics, Biochemistry, Polymer Chemistry. Polymers. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.